we're on. I remembered. I did not do a good job recording at the beginning, so I don't think I have the first 2 hours of content, but I have been recording since, so I will do my best. Keep me honest if I forget. We're going to do biochem part 4. I did have a request through the break to share some content about inheritance patterns. So I will for sure get that in the pathology section. So your 4th weekend, if I can slip it in a little bit earlier, I will. In the meantime, I'll remind me maybe if I don't post it by like Monday, send me an email and I'll post also a little like 5-slide lecture that goes through at least like the big inheritance patterns. But I would like to practice some Punnett squares with y'all. So we'll do that for sure last weekend, but I think we'll probably be able to sneak it in before then. So we will do it. All right. My goal is to get through this one and then to highlight some things that are in the popcorn, which is really just like meant for resources for you to kind of self-study and review as you see fit. So we're going to get through this and then we'll highlight some things in the last lecture, the popcorn lecture, and then we'll just open up to question time. And then if at that point, obviously you're all adult graduate students, you can leave whenever you want. But at that point, we'll kind of, if you want to take off, feel free. So probably be done with lecture content by like 4:15, and then we'll open it to kind of questions and popcorn topics. All right. Part 4. Lipid metabolism. So we've talked carbs, we've talked proteins, we've talked nucleic acids. Now we're getting into lipids to fats. There are 6 different classes. Fatty acids are by far, and triacylglycerols are the big ones we're going to talk about the most. Although we will mention and talk a little bit about metabolism of phospholipids, sphingolipids, and eicosanoids. We also will spend a decent amount of time talking about cholesterol and then the things that cholesterol can create. So that'll probably be our biggest chunk, is going through all the cholesterol hormone biosynthesis stuff. So we will do that in this section. When we're thinking about the body and where it's going to harvest fuel from first, carbohydrates are the easiest way for the body to get fuel because it has to break down the least amount of stuff, but it's the least high yield. It's 4 kcals per gram per carbohydrates. So if we have 1 gram of everything, we get the least amount of energy from carbohydrates themselves, but it's the fastest form for us to break down. So that's why the body utilizes it the most. Fat, we get the most, right? Fat, we get the most bang for our buck. But as we'll see, there's a lot more that has to go into breaking down fat. So that's why it's harder. The body doesn't like to do things that are challenging, even though there's a high reward. So sometimes we have to kind of trick the body into breaking down fat if we want it to. Protein, you get the same amount as carbs, 4 kcals per gram, but it's more challenging to break down proteins, and we can use proteins for more things in the body. So the body likes to not break down proteins if it can avoid it. And then alcohol, we're kind of in the middle. It's more like a quick source of sugar, but it gives us some negative byproducts. So the body doesn't love doing that all the time. When I mention fatty acid or fatty acyl, they're the same. So I will say them interchangeably throughout. So if I say fatty acid or fatty acyl, assume they are the same, the equivalent word. Fatty acyl is when we talk about it in like biochemistry, we talk about it as fatty acyl. When we talk about fatty acids in like nutrition, we say fatty acid. So, or in carbohydrate, or like in the heart or physiology. So you'll hear both interchangeably. Okay. So fatty acid-CoA, fatty acyl-CoA. Okay, so just kind of put those words together into one in your brain. So our fatty acyl-CoAs can get broken down or can get utilized or moved into 3 different kind of buckets. Energy via beta oxidation and ketogenesis, so creation of ketone bodies and breakdown of fats to actually create ATP. Storage in triacylglycerols or triglycerides, right? And then membrane lipids, actually utilized as, you know, a phospholipid or a sphingolipid or some type of like storage compartment. So those are kind of our big areas that we can utilize our fatty acyls in. When we look at the key kind of lipids or key fats and their chain lengths, we can look at obviously like these small, tiny ones, but really the important ones when it comes to kind of a bioclinical or biochemical sense is our linoleic and our linolenic acid, which are our essential fatty acids. So 18,2 and 18,3 linoleic and linolenic acids are going to be our important essential fatty acids that we need to obtain from our diet, right? And we'll look at them a little bit later, and we'll actually look at their chemical structure. Okay. So let's look at the difference between saturated and unsaturated. Saturated fatty acids have no double bonds. They are completely reduced. They can pack, not back, they can pack more tightly, and they have a higher melting point because of this. Unsaturated fatty acids get at least one double bond, carbon-carbon bond. They can be cis, which is typically our biological system. We see cis bonds or trans bonds. This is the configuration of the bond, not the identity of the unsaturated fatty acid, but the configuration of the bond. But we can see here the double bonds are often between carbons, and they're numbered from carbon to carbon. So you'll see like 5, 6, 8, 9, 11, 12. So they'll be numbered between these carbons. Double bonds between carbons can be also, they'll be numbered from, they can be numbered from the methyl end as well. So you could see methylated end 6, 7, 9, 10, 12, 13, 15, 16. All right, so triacylglycerol, or TAG, or TAG. We're going to see this glycerol backbone and these 3 fatty chains coming off of it. So our triacylglycerol TAG synthesis. So if we look here, we can see that the 2 kind of players or systems involved is going to be a liver and adipose tissue. So that's where we're getting these things from. The liver, we get glycerol. The liver and or adipose tissue is where we can get glucose from. So we're going to take both these things and use glycerol kinase, so with glycerol, or glucose, we can go to DHAP via glycolysis, and we can convert it to glycerol 3-phosphate, okay? From there, glycerol 3-phosphate, we get the addition of our fatty acids, so fatty acid 1, fatty acid 2, phosphatidic acid then is going to change kind of the structure. We're taking off a phosphate group, and then we're going to add in, we get our diacylglycerol. We then add in our fatty acid 3, and we get our triacylglycerol. And then from there, we can go either to the liver via the blood, via VLDL, or we can go into our adipose stores and store for later when we need energy. We'll kind of walk through this. Glycerol kinase is going to be really the rate limiting enzyme, because it's really the only major enzyme player that we haven't talked about yet. This only happens within liver and adipose tissue. So it's not happening anywhere else in the body, not in the skeletal muscle, not in the brain, only the liver and adipose tissue. Only the liver, though, possesses glycerol kinase. So only the liver can take glycerol and convert it into glycerol 3-phosphate. If we're in adipose tissue, we have to start from glucose or DHAP. So it's our glycolysis process. And then we'll see these linkages that are forming our ester linkages between glycerol and fatty acids using that oxygen in the normal carbon-to-carbon chain. Okay. So triacylglycerol synthesis, really think glycerol kinase from the liver, glycerol to glycerol 3-phosphate. From there, we add the fatty acid chains, and we end up with triacylglycerol, only occurring in liver and fatty tissue, nowhere else. Breaking down triacylglycerols, we can see here that our trigger to break them down, it's going to be upregulated in low insulin, high glucagon states. So if we need glucose, right? If we have lots of sugar, we're going to store fats. We're going to take sugar and we're actually going to convert it into fat for storage. If we're in a fasted state, we don't have a lot of glucose. We're in a hypoglycemic state. We need energy. This is a way we can harvest it. We can take our triglycerides or triacylglycerols. We can use this enzyme, hormone-sensitive lipase. It has to be phosphorylated to become active. And then we're going to see that it'll break off our fatty acids individually and our glycerol. So we get these 3 fatty acids and a glycerol, and they can all then be actually transported individually in the blood. As triglyceride itself, it can't just go into the blood directly. It has to be broken down into these fatty acids or glycerol form. You do need ATP to start the second messenger process to activate this enzyme. Okay. So that was triacylglycerols. But triacylglycerols require fatty acids, right? To create them, we need fatty acids. And then when we break them down, we have fatty acids. So we need to understand how we get fatty acids. They don't just exist in the world. So we're going to talk about how we create our fatty acids that then get combined together in threes to bind to glycerol backbone and then get stored as triacylglycerols. Fatty acid synthesis happens in cytosol of cells, in the liver and adipose, so two cells that we already know we can even create a triacylglycerol, and then also in the mammary glands and kidneys. So a few additional tissues can create fatty acids. So our fatty acid synthesis equation. So if we're going to make palmitate, which is a 16 carbon unsaturated fatty acid, unsaturated meaning what? Sorry? No double bonds? At least one double bond, right? Remember, if you had it, just reversed it. Remember, saturated, no double bonds. Unsaturated, at least one. We always want to say it in reverse. That's exactly why I asked that question. So unsaturated, no double bonds. So when we go to our fatty acid synthesis equation, we're going to make palmitate 16 carbon unsaturated fatty acid. We're going to need acetyl-CoA. We'll end up needing 18 of them in total because acetyl-CoA has 2 carbons. So 2 carbons times 8 of those molecules equals 16 carbons total. So that maths. We'll need then 14 NADPH. We'll need 7 ATP. We're going to run this 7 times essentially. And then we'll end up with palmitate, which is our fatty acid that we're creating. 14 NADP plus, 7 ADP, 7 phosphate, and 8 coenzyme A's. Okay. So if we look at this kind of overview here, and then we're going to kind of focus in a little bit closer. So if we look at this overview, we have glucose, citrate, acetyl-CoA, okay? Then we have moving down to malonyl-CoA, and then eventually palmitate. Well, we know it's not occurring this easily. There's lots of things that have to be involved, obviously, to get all the way down to palmitate, which is our final product, palmitoyl-CoA. In this process, we can see some regulation occurring. Citrate is going to be a positive regulator, an upregulator of acetyl-CoA carboxylase, which is this enzyme that governs the acetyl-CoA to malonyl-CoA reaction, which is like one of our first steps we have to do. Palmitoyl-CoA is going to have a negative feedback inhibition on this enzyme. So if we have our fatty acid, we're going to say, oh, we don't need to make any more. So slow this process. This enzyme, acetyl-CoA carboxylase, can become inactivated if phosphorylated. Okay. Insulin is going to upregulate this enzyme. So if we have lots of sugar in the blood and we don't need to use any more of it as glucose and we have all of our glycogen stores filled, we're going to say, well, what are we going to do? Let's make some fatty acids so we can make some triacylglycerols and we can store this energy for later. And so insulin then is going to be your upregulator to get this enzyme activated to take glucose, and it's going to make citrate and an acetyl-CoA. To pull that acetyl-CoA, instead of going through the Krebs cycle, we're going to say, nope, we're going to pull you out and we're going to instead use you to make our fatty acids. So step by step. There we go. So step 1, we have to get acetyl-CoA out of the mitochondria, because I said this process of fatty acid creation occurs in the cytosol. So we have to get it out of the mitochondria. So when we're looking here, we do glycolysis, whoop-de-doo, pyruvate. Pyruvate can get us to oxaloacetate and acetyl-CoA. And then we can get to citrate. Citrate's what can move out of the actual mitochondria. Then citrate can get reconverted back into acetyl-CoA. So citrate's your answer. So we convert acetyl-CoA to citrate. So we go through a couple steps of the Krebs cycle, and then we take that citrate, and instead of letting it stay in the mitochondria doing its thing as part of the Krebs, we actually move it out into the cytosol, and it gets reconverted into acetyl-CoA. And then we can use that acetyl-CoA in the cytosol to make our fatty acids. So our next step then, step 2, once we've got it out of the mitochondria, we have to get it to convert into malonyl-CoA. We'll see what that process looks like here in a second. So acetyl-CoA to malonyl-CoA uses acetyl-CoA carboxylase. This would be the enzyme I'd know of fatty acid synthesis, is acetyl-CoA carboxylase. It uses biotin as a cofactor. It does require energy input in the form of ATP, and it does take a carbon dioxide. And then we add that carbon dioxide essentially on the end of our acetyl-CoA molecule, to make malonyl-CoA. Malonyl-CoA has 3 carbons on it instead of the 2. And there's a coenzyme A attached. That's that CoA piece. Then we're going to do the linkage process. So then we're going to start taking these malonyl-CoA units, these 3 carbon units, and we're going to start to link them together. Okay. And we're going to use this via fatty acid synthase. That's the enzyme here that we're going to use. So here's our acetyl-CoA. Here's our malonyl-CoA. Here's our fatty acid synthase. Okay, so there's the players. Acetyl-CoA, malonyl-CoA, and fatty acid synthase. This is the reaction that we just watched. Okay. We're first going to reduce. So essentially, we have our fatty acid synthase. Acetyl-CoA is bound to it first. Malonyl-CoA gets bound to it next. So in this picture here, we have malonyl-CoA and acetyl-CoA on our fatty acid synthase enzyme. Okay. During this step here, from here to here, this is before we reduce, we're going to add our acetyl-CoA molecule to our malonyl-CoA molecule. That's our first step. So we now have our 1, 2, 3, 4 carbon molecule present on our fatty acid synthase. We lost 1 carbon in the form of CO2. So these are kind of the precursor behind the scenes steps that happen before we start this repeat process that's going to occur over and over and over again. So the first step is we're going to reduce. Then we're going to dehydrate. Then we're going to reduce again. And then we're going to move it over and repeat it. Okay. So we're going to reduce. So we go from here to here. This is a reduction because we can see our NADPH becomes an NADP. So this is getting oxidized. So this is getting reduced, this molecule. So we have a reduction first. We have an addition of hydrogen. There's another way we could say that, right? Then our next step, step 4, our dehydration. We remove a water molecule. So we dehydrate. We take this molecule, this water off, and we don't have water on anymore. We now have a double bond. We're going to then reduce again. We reduce this molecule again. We now eliminated the double bond. So we're back to that single bond. CH2, CH2, CH3. Again, using an NADPH this time. NADPH and NADPH. And then last thing we're going to do is we're going to move it over. So we switches places. We're going to move it from our here attached to this phosphate group. We're going to move it over here attached to this sulfur group. And then we start the whole process again. We attach our malonyl-CoA. We're then going to combine these 2 chains together in one long chain. And then we're going to reduce, dehydrate, reduce, move over. This process will occur until we have however many carbons needed to make our fatty acid. So if it's 16 carbons that we need to make our fatty acid, we're going to do this 7 times. Because the last time we're combining our last 2 molecules together, essentially, or the first time we start with 4, essentially, 4 carbons. So 4 carbons is our first time. Then we add 2 more, 6, 8, 10, 12, 14, 16. So we go through this cycle 7 times. 7 molecules of ATP, 14 molecules of NADPH, because we reduce twice per turn. So this is where I mentioned earlier, we're going to like memorize the processes, like what's happening in the reaction, but we're not going to memorize like actually what it looks like. So for fatty acid synthesis, you're going to do reduce, dehydrate, reduce, move. That's what you need to know. So we reduce, we dehydrate it, we reduce it again, we move it. Because it reduces twice, you know it's going to use 2 reducing molecules. So NADPH are your 2 reducers it's using. Your enzyme is fatty acid synthase. That governs this whole piece of the puzzle. Okay. And we're just linking carbon units together. Questions on this before I move on? This one trips people up, so I want to just pause. Malonyl-CoA, correct. Yep, so 1 molecule of acetyl-CoA is first added to fatty acid synthase. And then from there on is just malonyl-CoAs that are added afterwards. Yes. So acetyl-CoA, there's one acetyl-CoA that you'll take that gets bound to fatty acid synthase. The other acetyl-CoAs, all the other 7 acetyl-CoAs will all get converted into malonyl-CoA. And from there, those malonyl-CoAs will get added onto the acetyl-CoA. And we have to remember the malonyl-CoA is a 3-carbon molecule. But when we do our first step, that movement step, we lose a carbon as carbon dioxide. So malonyl-CoA gets bound as a 3-carbon molecule to fatty acid synthase. But when you combine the 2 carbon chains together, a carbon dioxide is kicked off. And so that's how you maintain that addition of 2 carbons each time. If you added an acetyl-CoA, I mean, in theory, it couldn't bind, so it wouldn't work that way. But you added acetyl-CoA, it wouldn't work because you'd only have 1 carbon each time that you're adding. Instead, you get 2 carbons using malonyl-CoA. You cannot build a fatty acid without using malonyl-CoA. You can't just use acetyl-CoA. Okay. Acetyl-CoA binds to fatty acid synthase. Then we get malonyl-CoA using ATP and biotin. We then bind malonyl-CoA with the acetyl-CoA to fatty acid synthase. We then combine the carbon chains together. We release a carbon dioxide in that. And then we start our process of reducing, dehydrating, reducing, moving. And then you repeat by starting again, adding your malonyl-CoA. So you reduce, dehydrate, reduce, move. Then malonyl-CoA, combine the carbon chains, reduce, dehydrate, reduce, move. Malonyl-CoA, move the carbon chains, reduce, dehydrate, reduce, move, until you've created the length of fatty acid that is needed, that's desired for this process. Palmitate, C18 is the most, or C16 is the most common. Okay, fun. Now, we were talking about, now what if we wanted to desaturate or elongate or change our fatty acid chain? Okay, so we made our C16, but we know that they're not all necessarily the same size, shape, and length. So you do have desaturases. Desaturases can add a double bond. They act in the endoplasmic reticulum, right? That's where we actually we make stuff in the cytoplasm. In this instance, we're making this in the cytoplasm. But if we want to change it anyway, we want to adjust it, it's going to go through the endoplasmic reticulum, and then the Golgi apparatus, and then it could be released, right? That's typical cellular machinery. So our desaturases, if we're going to change this fatty acid, it's going to have to go through some of those cellular machinery steps to have that adjustment made. Most commonly, a desaturase will act at carbon 9 on these carbon chains, but it can act other places. Humans do not possess a desaturase that can act beyond carbon 9. So we will not desaturate a carbon on a fatty acid that is greater than carbon 9. Okay. And how can you get like a double bond in carbon 14 or carbon 16? Because those are examples here. Well, you haven't added in those carbon chains yet. So you can get a double bond that's later down on the cycle, but you're going to have to add more carbons to that fatty acid to push the original bond over. So we can see in this example, we're desaturating, we're making a double bond. Then we're going to elongate with another malonyl-CoA. We're able to add that in. And then we desaturated it again, made another double bond. So we started with double bond at 12 and 19, and we had an 18 carbon fatty acid. And then we added a double bond at 6. We still had 18 carbons. Then we added in 2 more carbons. So we had now we have 20 carbons because we added another malonyl-CoA. And then we desaturated it again. So we have an additional double bond, but still 20 carbons. We can do as many adjustments as we'd like to for these molecules. Now, some aren't really biochemically utilized, so you're not going to have an endless supply of different types. But this is how we have a different array of fatty acids that occur inside the human body. Is using desaturases and elongation. That would be the extent I'd want you to know on this, okay? Know where it's acting. I don't think they'll ask you about the special carbons, but if you need to know, there you know. It does use an NADH, so it is an energy spending reaction to create this double bond. I could see them asking questions on this about location in the cell. I could see them asking a direct question about where in the cell would you see this process completed. I could also see them asking a question about cofactors, so NADH or niacin. So I could see them asking that type of a question. I don't believe you would see a question where they ask about specific carbons numbering. And I could see them asking about desaturation versus unsaturation, or monosaturated versus unsaturated, and those type of questions. All right. So when we look at our saturated to monosaturated fatty acid, CoA, you can see our saturated fatty acid here, and then our monosaturated fatty acid here. So we have no double bonds, and then we get a double bond. So our saturated fatty acid CoA, fatty acyl CoA desaturase. So this is just an example showing a specific saturase enzyme. And then we now have added our double bond in here. In doing so, we did use iron, FAD, and NADH. Just showing an example of what the cofactors could look like. Iron, riboflavin, niacin. So these type of questions. Anything that they could pull vitamins in, that's really where I think these questions could come. So we built them. Now let's break them down. This is beta oxidation. It's going to be another cycle. Okay, it was a cycle to build them up. It's going to be a cycle to break them down. So when we break down fatty acids, that process, fatty acid degradation, is called beta oxidation. So if you hear beta oxidation, that is the same as fatty acid oxidation or fatty acid degradation. So for beta oxidation, instead of reducing, dehydrating, reducing, moving, we're now gonna do oxidize, hydrate, oxidize, cleave. Very similar, just almost like reverse process, right? So we look here, we're gonna be in the mitochondrial matrix this time. We have our fatty acyl CoA. We're gonna first oxidize using acyl CoA dehydrogenase. This would be your rate limiting step, this first step. Then we have a hydratase, so we're going to hydrate by adding water. Then we have another dehydrogenase because we're doing another oxidation. And then we're going to break it apart using a thiolase, beta-ketothiolase. Okay. And then it's going to repeat itself until we've broken apart all of our fatty acyl, their whole fatty acid, down into acetyl-CoA. Okay. So rate limiting step would be your first enzyme, acyl-CoA dehydrogenase. Oxidize, hydrate, oxidate, cleave. That's your process here. So if you know, if you can if you can put in your brain fatty acid synthesis, reduction, dehydration, reduction, move using malonyl CoA. Beta oxidation, oxidase, hydrate, oxidize, cleave, get acetyl CoA. You'll be pretty well set for most of these questions. Fatty acid synthase to build your fatty acids, acyl CoA dehydrogenase to break them down. Different compartments. So you can see here, this is how we would get so much energy generation. Do not memorize this, do not use this. Just know this is your explanation of why fats are so energy rich, right? So for all of this, if we break down a 16 carbon and palmitate fatty acid, we're going to end up with 14 FADH, 21 NADH, and 96 ATP. Okay? Because each acetyl-CoA provides 12 ATP when converted into carbon dioxide and water by the TCA cycle. So 16 times, or like, what is that, 8? Yeah, 8 times 12 is 96. So if you're comparing glucose to one fatty acid is going to give you a heck of a lot more energy. But the body has to do more steps, you can see, to break that down. So the body is less likely to want to do that because glucose is just so easy. Just churns it through glycolysis. Well, a question comes up from time to time. I don't say this as much on NPLEX, but I've had students ask me, what if the fatty acid is odd change? What if it's not an even number? We've been talking about 16, 18, 20, ends up in acetyl-CoA. What if it's 13, 15, 19? If that's the case, there is a separate pathway that you can go through. So this here would be your final breakdown. You would go through this process, beta oxidation, until you end up, instead of having just acetyl-CoA, you'd have a bunch of your acetyl-CoAs and then one weird 3-carbon molecule, which is your propionyl-CoA, your 3-carbon attached to your coenzyme A and your sulfur. This can get converted to methylmalonyl-CoA, and it uses this carboxylase. I wouldn't memorize the enzyme, but it does use biotin and bicarb and ATP. It uses a lot of energy. It's an ATP to AMP 2-phosphate. Methylmalonyl-CoA can easily change its isomer to either D form or L form, and then L form can get converted into succinyl-CoA. And succinyl-CoA can move into the Krebs cycle from there. And remember, beta oxidation is occurring in the mitochondrial matrix. So this can then get pushed into the Krebs cycle and be used as an intermediate. This does require coenzyme B12, and that does mean it needs B12 to move forward. Okay. So thinking about metabolism as a whole, fatty acids, they travel in the blood from either gut or adipose tissue. They're attached typically to an albumin carrier in the blood, and then they can get into the mitochondria via our carnitine shuttle. So the carnitine shuttle is a shuttle for our fatty acyls or fatty acids. Carnitine is generated from lysine and methionine amino acids, and the biosynthesis of it occurs in liver and kidneys. You may or may not. Some sources say you need vitamin C, some sources say you don't. For NPLEX, I would say you do. So for NPLEX, just assume sure you need it, but I've seen sources say it's not required for carnitine synthesis. From here, your fatty acids that are inside the cytosol can become a fatty acetyl-CoA by adding CoA, using some energy, using acetyl-CoA synthetase. Fatty acetyl-CoA can move through the outer mitochondrial membrane inside the mitochondria. From there, the acetyl-CoA group is bumped off and you are bound to carnitine. So you get fatty acetyl-carnitine. That can then move into the inner mitochondrial membrane. And then fatty acetyl-carnitine is broken back off. The carnitine is removed and recycled, and the fatty acetyl-CoA is now on its own again. It can undergo beta oxidation. So that's how that transport happens. Kind of like the malate shuttle, kind of like all these shuttles, they're essentially using another molecule to do some type of cyclical process to move from one chamber to the next, to move inward. Okay. So taking a step back, we've been talking about fatty acids and we talked about triacylglycerols. But I want to just put into our perspective what these things look like and kind of the comparison of these fats overall. We're not going to spend a lot of time on these other fats. I just want in general, you'd have an idea what their structural makeup is. So a triacylglycerol is easy. It's our glycerol backbone with our 3 fatty acids. We know how to make this. We know where this comes from, our 2 sources, and we've now created fatty acids from scratch. So we know how to create our triacylglycerols. This is going to be our blood lipoproteins. So the things we measure in our cardiovascular disease, as well as our adipose stores. And this would count as a glycerolipid. So that's a category of fat. If we then wanted to look at, okay, well, what is a glycerophospholipid? It's taking components of the glycerolipids and components of phospholipids. So it's kind of coming from both sources here. And there's a ton of them. Ones that we might know and love, phosphatidylserine, phosphatidylcholine, so things that we actually administer as therapeutics for patients. The difference is this third chain isn't a fatty acid, it's a phosphate and whatever the head group is. So phosphatidylserine, so this is going to be a serine group to a phosphate. Phosphatidylcholine, a choline group bound to a phosphate. So that's your difference here with your glycerol phospholipids. Your ether glycerolipids, really the one that we think about is like platelet activating factor. It's the only kind of infamous one. It would count as either a phospholipid and glycerolipid category. And the difference here, it looks like a glycerophospholipid, but instead of having a top fatty acid, it's a glyceroether. So this chain, it's not a glycerol chain on its own. It's combined through this oxygen to a carbon chain over here. sphingolipids. So we have sphingofospholipids, so sphingomyelin, or myelin. Okay, sphingomyelin. That has sphingosine, which is its own special compound. We'll look at that here in a minute. Bound to a fatty acid and a polar and a head group. So this looks the same, it's just the backbone's different. And then our glycolipids. This is a sphingolipid, has a carbohydrate, hence the glyco, a fatty acid, hence the lipid, and then a sphingosine backbone. Okay. Why do we care? I've seen them ask questions kind of putting things in categories or boxes, or at least having name recognition is huge on NPLEX. Like, I've seen that word before. Where can I place it? Fats. Great. Like, at least then you're in the zone. Hydrophobic, water-fearing, fatty acids, high energy. You can kind of put yourself in that place when answering questions. I do not want you to memorize the next 4 slides. Okay, I just want to talk through it just so we can have an understanding of what sphingosine is and sphingolipids, and we'll move on. Okay, so ceramide helps create sphingosine, or it comes from sphingosine and helps us get to sphingomyelin. Okay, so we're talking about these sphingolipids, and we need to understand what sphingosine is because it's something new. So that's why these slides exist. It's just to explain what this molecule is. So sphingosine is a 16-carbon amino alcohol. It has an unsaturated hydrocarbon chain. And then ceramide is sphingosine plus a fatty acid bound to it. Okay, so ceramide, sphingosine, this, plus a fatty acid. So it's essentially this like F, okay, without the polar head group. So that's what we're looking at here. This ceramide molecule can go utilize phosphatidylcholine and become sphingomyelin. It can be created into these ceramide forms. Over here, a ganglioside form. So this is really the starter molecule for our sphingolipid category, is this ceramide. The breakdown of sphingolipids really more gets exciting when we talk about like Tay-Sachs or Gaucher's disease. Again, neither of these are on your NPLEX, so I don't care that you know about them right now. I just want you to have like place awareness. When we break down sphingosine and then break down, we'll see like this is the creation, serine and palmitoyl-CoA, sphingosine, ceramide. This is where we started from the last slide. We can get sphingomyelin, cerebroside, and then we can break these back down into sugars and into fats. So we can kind of circle them back on themselves, or we can go and break them down into these individual components. Sugars can come in and go out. So there's a lot that we can do with this ceramide molecule. It's really all I want you to care about from this slides. I just labeled reaction oxidation. I was feeling excited, my little things. In certain conditions, we can see there's defective enzymes. These are the enzyme deficiencies, the diseases, and then the accumulated lipid in question. I don't believe these diseases are on your NPLEX I. They could be on NPLEX II. So this is more of a reference sheet for you in the future. Okay, moving on. Cholesterol time. Isoprenoids. All right, isoprene is a 5-carbon unit. I gave you a little picture. We can find terpenes in essential oils of plants. They have tons of isoprene units. When we get to the exciting things in the body is when we get to triterpenes, just 6 isoprene units, which is our steroid, okay? Cholesterol is our precursor to steroid hormones, including vitamin D, and it's also found in our cell membranes, our lipoproteins, and it's a precursor to bile salts. So it's pretty important. And we're going to talk about how we synthesize it, and then we'll take a break at 4, and then we'll come back and finish up this lecture. So cholesterol synthesis mostly occurs in the liver, cytosol. The important things to note is HMG-CoA synthase and HMG-CoA reductase. HMG-CoA reductase is your rate-limiting step, but HMG-CoA synthase is an important step as well. Acetyl-CoA and acetoacetyl-CoA combine together to form HMG-CoA using HMG-CoA synthase. Then we reduce it and we get mevalonate, okay? Mevalonate using HMG-CoA reductase. Then a bunch of steps happen and we get cholesterol. I don't care about those for this exam. Care about the first 2 steps. That's it. Once you get to mevalonate, you're good. You're good to go. Okay. So we can see here this process, acetyl-CoA is removed from mitochondria by the same mechanism in fatty acid synthesis. We use that citrate shuttle. HMG-CoA reductase is our rate limiting step. So this is the rate limiter here. It is going to be down-regulated with cholesterol. So we have negative feedback inhibition. Down-regulated with statins. It's a structural analog, so blocked. Down-regulated with glucagon. It promotes inactivation via phosphorylation. And upregulated with insulin. This will promote dephosphorylation. So it'll allow it to be activated. But we can see acetyl-CoA getting converted to acetyl-CoA. So we have 4 carbons now. And then we see this conversion here with HMG-CoA synthase. We've added in essentially this acetyl-CoA molecule. And then we get our HMG-CoA reductase. So we undergo this reduction. And now we have malabonate. Cholesterol can form into a cholesterol ester. This is more hydrophobic, and this can be helpful for transport in lipoproteins. They do this by using our ACAT reaction, which is our acetyl-CoA cholesterol acyl transferase. Not super important that you know this enzyme. Just note that if you hear about a cholesterol ester, it is a thing. I think a lot of times name recognition and NPLEX is what gets us. We don't know it, so we think it's not real. So it is real. It makes them more hydrophobic, and it's helpful to transport them within a lipoprotein. Okay. Let's synthesize some steroid hormones. All right, so the student version, I have this slide here, and then I have another slide that says practice writing out these pathways. And the PowerPoint version, you'll see like I've wrote them all out for you. So, but I encourage you on your own, it's really high yield to really know this picture and know the different pathways and the endpoints. These enzymes are great NPLEX test questions, and great meaning they're horrible, but they are asked about on every NPLEX exam. Okay, so we're starting with cholesterol, and we're going to go kind of one by one. We're going to focus in on these areas. Recognize our endpoints. So we have cholesterol, our key pieces: cholesterol, pregnenolone, progesterone. We have aldosterone. We have cortisol, testosterone and DHT or dihydrotestosterone, and then our estrogens, E1, E2, and E3, or estrone, estradiol, and estriol. Okay, so these are the endpoints that I want us to be able to walk through how we get there. So first we have to get to progesterone. This is the shortest one, so which is nice. So we're going to get to our progesterone molecule here. So we start with cholesterol, and then our first precursor really is pregnenolone. Pregnenolone then is going to get to progesterone using 3-beta-hydroxysteroid dehydrogenase. We will abbreviate this as 3-bHSD. So 3-beta-HSD. That gets you to progesterone. Easy peasy. Only one pathway to get there. Pregnenolone is your intermediate. Our aldosterone synthesis, we're going to follow the same track. So we now have gotten to progesterone. We next need to go from progesterone to deoxycorticosterone, and then to corticosterone, and then aldosterone. So to get from progesterone to deoxycorticosterone, we're going to utilize 12-hydroxylase. So we're essentially adding a hydroxyl group to the 12th carbon. And then to get from deoxycorticosterone to corticosterone, we do 11-beta-hydroxylase. So again, we're adding another hydroxyl group. And then to get corticosterone to aldosterone, we do aldosterone synthase. So we've created an oxygen double bond. Okay. Yeah. For progesterone to decorticosteroid, it's 12-hydroxylase. Or 21. Wait, did I say 12 or 21? Oh, thank you. Typo. Yes. Sometimes I get dyslexic. Sorry. It happens to me. Okay, here we go. There, 21. Thank you. Keep me honest, folks. The only way. Okay. All right, cortisol synthesis. This one is probably, if you had one that got tested on every time, it'd be this one. Okay. I think every time I've seen this test, there's been something about cortisol synthesis. So with cortisol synthesis, we can have two ways of getting there. We're going to start with cholesterol to pregnenolone regardless. And then pregnenolone can either get converted into progesterone or it can get converted into 17-alpha hydroxypregnenolone. So you see it can split. It can go this way or it can go this way. So pathway 1, we're going to go down and over. Okay, pathway 2 will go over, down, over. So pathway 1, pregnenolone gets converted via 17-alpha hydroxylase to our 17-alpha hydroxypregnenolone. Okay. Then our pregnenolone is going to get converted into 17-alpha hydroxyprogesterone. Uses 3-beta hydroxysteroid dehydrogenase. Pathway 2 has the enzymes just in reverse order, right? Same enzymes, reverse order. First, in pathway 2, we go pregnenolone to progesterone. So we're using 3-beta hydroxysteroid dehydrogenase. Then we're going to convert progesterone to the 17-alpha form using 17-alpha hydrolase. So you see we have the same enzymes. There's no enzyme change. It's just the order we use them and the intermediates that happened. Oftentimes NPLEX questions will be like, if this enzyme is blocked, what's happening to this end product? So if you know there's multiple ways to get there, you just need to know where in this pathway is the enzyme blocked. Is it only, this is the only pathway, right? If it's in the instance of cholesterol to cortisol, if we block 17-hydrolase, alpha hydroxylase, we know we need that for both pathways. So we're not going to be able to get there either way. So cortisol, we're not going to be able to synthesize cortisol. So if they're asking what would happen to cortisol synthesis, it would go down. If they're asking what would happen to progesterone synthesis, if we block 17-alpha hydroxylase, well, then our pregnenolone is going to all be going to progesterone. That's all we can do. So that's going to go up. So you can start to see how you can play with these questions, depending on where you're blocking and depending on what they're asking about, to see where things are going to be shunted to and where things are going to go. So now, we've gotten here, 17-alpha hydroxy progesterone, we have 2 more steps. This is the joint steps. We're going to convert 17-alpha hydroxy progesterone to 11-deoxycortisol. We're using our 21-hydroxylase. And then we're going from 11-deoxycortisol to our cortisol using 11-beta hydroxylase. I'm not talking about where we're making things happen yet. We're going to get there, okay? We're just talking about the enzymatic, the micro, the molecule steps to get to these places. Okay. DHEA, dehydroepiandrosterone. How do we get here? We go straight down, one way. Pregnenolone, 17-alpha hydroxypregnenolone, DHEA. So we're going to go through cholesterol to pregnenolone. Then we are going to go from pregnenolone to 17-alpha hydroxypregnenolone using the same enzyme, the 17-alpha hydroxylase we previously used. Then the new player on the block is 17-20 lyase. That gets us to DHEA. Okay. Now testosterone. We're just opening back up into this world. To get down to testosterone, or dihydrotestosterone, DHT, we go from cholesterol to pregnenolone, and then we have 4 options, all using the same enzymes. Okay, so this is a kicker. All options will at some point need to use all the following enzymes, but the order is what's going to change. Okay, so the enzymes that we have to use is 3-beta hydroxysteroid dehydrogenase, 17-alpha hydrolase, 17-20 lyase, and 17-beta hydrosteroid dehydrogenase. Okay. It just depends on which way we go. So I'll show you our pathways here. We can go pregnenolone here, 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 here. That would be going 1, 2, 3, 4. Or we could change our color. We could go pregnenolone, 17-alpha, DHEA, androstenedione, and then testosterone. So that would be 1, 2, 3, 4. See, starting to see my drift now. Or we could go pregnenolone, progesterone, 17-alpha, androstenedione, testosterone. So we could start here, 1, and then 2, 3, 4. Or just to make it even more fun, we could go down here, over to here, down here, and then down here, a little like L action. That'd be 1, 2, 3, 4. I think I got all the combos. So needless to say, lots of ways to get to testosterone. Go androgens. Okay, so we can get there. What's going to depend on like where and why and how is the location of what's happening. That's going to affect things because these things happen in different locations. Also, obviously, enzymatic concentration, our substrates and products, what's already formed in high amounts. Are we having backlog one direction or the other? That could cause us to push backwards and not go down certain pathways. So that's what we'll decide. But either way, these 4 enzymes are used, regardless, from cholesterol. From testosterone to DHT, it's super easy. It's one enzyme, it's 5-alpha reductase. Easy peasy. That's what takes testosterone to DHT. DHT is a really hyperbinding form of testosterone, typically used, used multiple places, honestly. So we'll see this, especially when we talk about our physiology section, when we talk about hormones. We'll lean into this in its action more. And last, our little estrogens. Our estrogens, E1, 2, 3, and 4. We have to get to androstenedione or testosterone first. So we have lots of ways we can get there. But once we're here, we're going to use aromatase to convert either into E1 or E2. So androstenedione can convert using aromatase to E1, which is estrone, or testosterone can be converted using aromatase to estradiol, which is E2. Estrone can become E2 or E3. So estrone, which is E1, can be converted to estradiol or estriol. Estradiol can only become estriol, E3, and estriol is its final form. Can't become any other estrogen. I encourage you to play around with that picture, that main picture, work through pathways, say if this concentration's increased, what would happen to this product over here? Yeah.
Did we even think about physical structure?
No. I wouldn't even care about the physical structure.
Write this out, just do the words.
Do the words. Unless if you are a chemistry-minded person, the physical structure helps you, I would just do words and abbreviations even for most. Because most of those, if you use abbreviations, you'll be able to, and you work on memorizing via abbreviations, you could work back to a full word if they gave you a full word. Yeah. Yep. So now the locations. Initial synthesis of these things takes place in the adrenal cortex. So we remember our favorite, GFR, salt, sugar, sex. Okay, that is the mnemonic. So our zona glomerulosa is our mineralocorticoids, that's our salt. Our zona fasciculata is our glucocorticoids, that's our sugar. And our zona reticularis is our androgens, that's our sex. Okay. So mineralocorticoids, aldosterone, glucocorticoids, cortisol, androgens, testosterone, DHT, and then estrogen. What about adrenal medulla? The adrenal medulla produces your catecholamines. So epinephrine comes from your adrenals, but your adrenal medulla, not your cortex. Super good NPLEX question. There is a different embryologic difference. Okay, so our neural tissue comes from our ectoderm, I think. Don't fully, let me make sure. Yep, mesoderm, no, endoderm, glands. Yep, so our ectoderm is our neural tissue, and that's going to give us our adrenal medulla, which is going to be more of our neural connection, right? Because when we have that autonomic response and we want to send out norepinephrine and epinephrine for our sympathetic nervous system response, the norepinephrine is our neurotransmitter form. The epinephrine comes out as a hormone via the adrenal medulla. So it's that neural connection. Yeah, good call. I would then assume to say, I want to jump to say that the medulla or the cortex then, because it's glandular, is endoderm, but I'd need to check. It could be meso. I'd need to confirm. Yeah, but most glands are endoderm, but I would, again, let me look. Yeah, no, it's good. My embryologic brain is not quite there yet. Cortex, mesoderm. Thank you. Is that Gigi? Yeah. Thanks, Gigi. You're the best. Neural crest. Yes. Okay, good. Ectoderm. Awesome. I love it. All right, let's break one last time. Let's take a little break. Unless you, well, okay, I'll give you an option. Break or power through. Power through and then stop. Let's power through and stop. We got this. Let me see what I committed us to here first. Oh yeah, let's power through. This will fly, so we're good. Let's power. Okay. Steroid hormone breakdown. So we break it down. 95% of cortisol and cortisol metabolites are conjugated in the liver with glucuronic acid and sulfate and excreted. So just keep that in mind. Liver is where we're actually doing this conjugation, this breakdown, this elimination. But cortisol elimination, this is really one of the steroid hormone breakdowns that we get asked about. Cortisol can get converted to cortisone. We can see these interconverted. Cortisone is the inactive form. Cortisol is what is the active form. Then to break it down further, to kind of eliminate from the system, we use either 5-alpha or 5-beta reductase. 5-alpha reductase for cortisol, 5-beta reductase for cortisone or cortisol. That would be your enzyme of choice to know. Otherwise, I would not worry about the rest of the processes in this breakdown. 5-alpha reductase. This is just showing kind of where we would go. So 11-beta HSD2 going to from cortisol to cortisone is more active in kidney, colon, saliva. HSD1 going here is active everywhere else, really. Skin, liver, adipose, lung, muscle, gonads. So all of your places where you would really see cortisol affecting you, your body. The body. Okay. It's also used in that conversion of testosterone to DHT, that 5-alpha reductase. So when we actually take a step back and look at digesting our fats, because that's where we started this conversation, we're going to take in triacylglycerols, RTGs, we're going to bring them through the stomach, get them into the small intestine, and that's where our liver is going to create our bile, which gets stored as bile salts in our gallbladder and excreted into the small intestine along with bicarb, lipase, and colipase. These then three kind of components are going to take and with our bile salts are going to circle around and bind up our triglycerides, break them down into fatty acids. These fatty acids are going to be essentially broken down further. They're going to be combined with our bile salts again in these little micelles. And then from here, we'll see the fatty acids themselves are taken up into our cells. The bile salts are either excreted or they're recycled. And then from here, our fatty acids are converted back into triglycerides, and then they're going to be packaged into chylomicrons, which then can go into the tissue and into the blood. So micelle, small intestine, into our brush border cells, or brush border epithelium, and then we're going to break it back down into triglycerides. Triglycerides then get packaged in chylomicrons, which goes into the blood and can travel then throughout the body. They'll go to the liver. Okay. The enzymes that we care the most about, our pancreatic lipase is the big one. That's going to be the degradation of dietary triglycerides in our small intestine. Lipoprotein lipase, LPL, breaks down circulating triglycerides and that are in chylomicrons and VLDLs. Hepatic triglyceride lipase is the degradation of any triglyceride left in IDL when it gets into the liver. And then hormone sensitive lipase is degradation of triglycerides stored in our fat cells, adipocytes. So these are your big ones. Pancreatic and lipoprotein are the two ones I see tested on the most. Okay. Our bile salt formation. Synthesized in the liver. Don't let them pull you to the gallbladder. Don't do it. It's stored in the gallbladder. Synthesized in the liver, uses cholesterol. 7-alpha hydroxylase is your rate limiting enzyme. You're then going to get the conversion to 7-alpha hydroxycholesterol. And then you get the conversion into these intermediaries, which finally get you down to chinocholic acid and cholic acid, which are your initial bile acids. Okay, so that's your end products here. The pKa of bile salts is around 6, so it's a pretty good buffering. Like I said, 7-alpha hydroxylase is your rate limiting step. You then will see these cholic acid and chinocholic acid get conjugated with amino acids. This allows a lowering of pKa of bile salts to 2 to 4. We think about the fact that like literally hydrochloric acid has been coming into our intestines. It's really helpful to have this pH a little bit lower so they can actually act and bind and interact with these components that are coming out into the small intestine from the stomach. So we'll see conjugation. You'll have an improved amount of solubility and detergent action if bile salts are conjugated. If they're not, they can still work, but they work much better if they're in a conjugated form. So you can see taurine and glycine getting conjugated here, and you get taurocholic acid and glycocholic acid and the different pKa's. That does require energy, so you are using ATP. So if we look at our primary bile salts, it's the ones that are formed directly from cholesterol. Our secondary bile salts are are deoxygenated forms. So we have a deoxycholic acid and a lithocholic acid. Their bile salts are deconjugated and dehydroxylated at your position 7 by gut bacteria. So gut bacteria is what dehydroxylates and deconjugates. Our dehydroxylated bile salts are what we call our secondary bile salts. So that's what these are here. Secondary bile salts are less soluble. They are less likely to be able to be reabsorbed and recycled back into circulation. The liver can reconjugate, but they can't rehydroxylate a bile salt, a secondary bile salt. So once they reach this state, a lot of times they're on their way out at this point. And so then the body's going to make a replacement. The body really tries hard to recycle bile salts as much as they can because it takes a lot of energy to make cholesterol. And so we don't like eliminating cholesterol to make bile salts. We really like to keep our cholesterol as much as we can. A lot of energy to create that molecule. So we recycle 95% of bile salts per day. Only 5% or less go in our feces. Okay. Our lipoproteins, this is where everyone in cardio like collectively passes out and doesn't remember, and that's okay. I don't too half the time. So our lipoproteins from our liver and our intestine, we generate HDL, which is the lowest amount of triacylglycerol, so the lowest amount of triglycerides, and the highest amount of, and a higher amount of cholesterol. Its goal, HDL, is to deliver cholesterol from peripheral tissues to the liver for elimination, right? It's our good cholesterol, our beneficial cholesterol, our happy cholesterol. VLDL gives us LDL, which is composed of not a lot of triacylglycerols, but more than HDL, the highest amount of cholesterol total, right? And this delivers cholesterol to the peripheral tissues and to the liver. Our liver generates VLDL, right? So our liver generates VLDL, which will generate LDL, right? So we kind of had this backwards. Our liver generates VLDL. This has the most amount or the greatest amount so far of triacylglycerols, a low amount of cholesterol, not much cholesterol in VLDL. And this delivers endogenous triacylglycerols to peripheral tissues. And lastly, our intestine makes our chylomicrons, right? We saw that earlier. This has the most amount of triacylglycerols, tiny little bit of cholesterol, And this delivers dietary, so exogenous triacylglycerols from our food to our peripheral tissues and our liver. So I would know these four: HDL, LDL, VLDL, chylomicrons, know who created them, know the ratio between triacylglycerols to cholesterol, so who has the most, the least, etc., and what their function is. If you know that, like if you know this slide, you can answer almost all cholesterol mechanism questions. Yeah. Protein content in each of these? Oof, no, not off the top of my head. I'm sure we could find it out, but I do not have that off the top. That feels right. I like that. I'm going to go with HDL having the highest. I will confirm for confirmation's sake, but that feels correct. If we look at some of these structures as a whole, we can see our LCAT and our CETP. LCAT is lecithin cholesterol acyl transferase. It catalyzes the esterification of cholesterol. Remember, making it more able to be carried in these different molecules. Our cholesterol ester transfer protein allows for transfer of cholesterol esters to other lipoprotein particles. So this is really just the two enzymes that help cholesterol be transported. And then we can kind of look at the collection of these different pieces. And then some of these proteins, I have them listed here. So our apolipoproteins, E, A1, C2, B48, and B100, talking about what their major function is. So activating LCAT is A1, binds LDL receptors B100, chylomicron secretions B48, and then you can see where you would find these proteins in existence. So HDL having a lot. I mean, I'm sure concentration-wise, this doesn't say concentration. This just says who you would find where. The least in LDL. My only thing to say about eicosanoids, it really comes into play more when we talk about inflammation, like inflammatory pathways in the body. But when we take in our fatty acids, we see we have linoleic, arachidonic acid. Arachidonic acid is kind of our big backbone for where we get into when we start talking about prostaglandins, leukotrienes, thromboxanes, all of these kind of anti-inflammatory or inflammatory molecules. So when we go down this pathway, arachidonic acid can become a membrane phospholipid, or we can have arachidonic acid moving down into prostaglandins, moving into leukotrienes, or moving from prostaglandins to thromboxanes. We will talk about all three of these molecules when we go into immunology and micro. So in 2 weeks, we will go through what are their mechanisms in regards to inflammation, in regards to the immune system, etc. We're just doing synthesis for today. This one piece, though, this is exciting because it does show your COX enzyme, your cyclooxygenase. And this is where when you take an NSAID, this is where it's decreasing or blocking or downregulating this conversion. Okay, this is just talking about freeing the phospholipids from the membrane. I would say lower yield overall, but if you want to look into it, you can. This is the picture that I would focus more on for arachidonic acid. It just looks at if we have cell membrane phospholipids and they're released from the cell membrane, where would we go and who's involved? Who are some of the players? So we have our cyclooxygenase enzyme that can get us to our prostaglandin 2s to prostaglandin H2, and then our actual prostacyclins, which cause vasodilation and inhibit platelet aggregation, or our thromboxane A2, which causes vasoconstriction and promotes platelet aggregation. So either end of the coin, or we can create directly our PGD and PGEs, which causes vasodilation and increased vascular permeability, allowing cells to move in and out of the blood vessel. Again, more immune activity. Our lipooxygenase from our lipooxygenase enzymes, so we kind of do this 5-HPEAT conversion, which can either give us our leukotrienes or our lipoxins. Lipoxins are inhibiting chemotaxis and neutrophil adhesion. And our leukotrienes are going to cause our vasoconstriction, bronchospasm, and increased vascular permeability. So really, inflammation. We'll look at this and their functions more in 2 weeks. Our last two, Omega-6s, linoleic, arachidonic acid. Linoleal-CoA goes through desaturase. Linoleal-CoA goes through elongation. Linoleal-CoA goes through desaturase, and then we have arachidonyl-CoA, which will then eventually become arachidonic acid. But we need our omega-6s, our linoleic. Omega-3s are EPAs and DHAs. So linolenic, omega-3 fatty acids, desaturates to a steroidonic acid. We get elongates, then we desaturate, we kind of do some stuff. And then we get our EPA. We elongase, desaturate, oxidate, and then we get our DHA. So when we talk about consuming omega-3 fatty acids, they're either in the EPA or DHA form. These are the forms they're talking about here. So if we start with linolenic, our ALA, and then we can convert to EPA, and then we can convert to DHA. So that's your conversion order. That would be the most I would kind of know in this process. You did it, folks. Wow. A lot of biochemistry in a day. I am just going to show you what this has in it. This is your popcorn. This is your choose your adventure for your own self-study. I just want you to have these resources. This is where I listed out all your vitamin stuffs that wasn't kind of already included. It's a nice way to get a summary page. So I do have the summary of the vitamin, the other names, the active form, and the function, and if they're water or fat soluble. This also includes a lot of enzymes that they're involved in as cofactors. Individually, each slide also has their own version, their own slide that talks about what its function is, what its active form looks like, where it could be found, and the processes and the enzymes. So pyruvate dehydrogenase in pyruvate oxidation. So these are all the places, the major players where you find thiamine B1 as a cofactor. So I hope this helps you have to do less of your own self-harvesting. What I would encourage you do is as you draw out your pathways, as you uncover these enzymes on your pathways, write the cofactors there as well. And that way you can really integrate this knowledge. If an enzyme's on this list, it's probably a rate limiting step or an important enzyme. So it'd be one that you should try to find on your pathway. So I would have a pathway that's unlabeled with no enzymes, no cofactors. Look through this list and find where these enzymes in this list go, and then put the enzyme name and the cofactor there. Again, structures don't matter. No structure, you will never see a structure on NPLEX. You'll just see words. Okay, so I have them all for all the water solubles. I also have them for their fat solubles too. For the fat solubles, I try to show some synthesis pathways as well. So if you're like, how the heck is vitamin D synthesized and what are the forms? Got you. Vitamin A, same. And vitamin K. I also have some on here for your minerals. So they're not as complete, but I have some of them as well. And then if you're an overall person, this is your overall vitamin summary. So kind of where are the vitamins in general in these pathways? This is your glycolysis, your Krebs. I think this is your urea cycle over here. Your fatty acyl cycle over here, your triacylglycerols over here. So you can also utilize this, unlabel the vitamins, and add them back in for self-study. If you're feeling really excited, want to go through genetic SNPs, I mean, go to town. I think this is low yield study at this point. It's trying to memorize all the important genetic SNPs. I would say when we get into path, know the major players in pathology, but don't spend a lot of time on this. It's just not worth it. Not worth it. I have, if you're interested in how amino acids specifically are breaking down, or where we find things like GABA or catecholamines or indolamines. If you get stuck on a question, I probably have an answer in this PowerPoint, biochemistry wise. So as you're going through practice questions, if you're like, wait, how do catecholamines, what does that pathway look like? It's probably here, so you can search it. The few pathways that we didn't touch on that I do think you need to go through on your own for some self-study would be the pentose phosphate pathway. This pathway is small but mighty because it generates our NADPH. So that's the function, is to get NADPH back. So I would say spend a moment at least looking at this process. It's on all your major summary pages. it's involved. It's listed as a little circle, but when we deplete our NADPH, the pentose phosphate pathway is how we return, how we get it back. And then I also have every pathway that needs NADPH. You can kind of see, oh my gosh, these are all the different pathways that deplete it or that use it. So this is where pentose phosphate pathway is working for. If you're still not sure about like glutathione, oxygens, free radicals, I have all those things like respiratory bursts where they're all used, our antioxidant harvesting. So feel free to utilize those as you see fit. Same with ketone synthesis and breakdown. And then I wrap up with kind of some of these big pictures that you could use to like hide things and self-study and add things back in. This is carb lipid metabolism combined. And then this is your rate determining enzymes of all your major processes. So one nice sheet, and then the regulators, not cofactors. Another summary sheet as well. So really this is just meant to, I hope that you have like so many biochem resources that I want you to not highlight, not read. I want you to practice question, test yourself, write things out from memory, and then check your answers. That's going to be your best high yield for the next month. What questions for me about how to approach this, where to go from here, what you should be doing next? As much as you can at this point. Yeah. The question was, how much time should you spend each week? As much as you can fit into your schedule. I mean, I could give you a number, but you're in school, right? So as much as you're able to, this is your push. Bye. Thank you. See you next week. I'll probably see you on campus. But it's 8 hours every day. That's like the study schedule, like 40 hours a week, right? You're not going to necessarily do that. It's just not possible with school. So as much as you can fit in. When you're in between classes, when you're, your first 4 weeks hopefully aren't super duper test heavy yet. So I'd say like rob Peter to pay Paul a little bit and study on this more than maybe your actual block courses, as much as you're able to. Okay. Awesome. Any online, yeah, I see a chat question. The non-Blake sides are on, the non-Blake sides are on Moodle. So you can find all the non-Blake sides. The PDFs are of the things that have blanks, and then the PowerPoints below have everything filled in. So you do have access to all of those. And then the Biochem popcorn doesn't have any blanks at all. It's just all filled in currently for you. So you can create your own blanks if you want to. But you have access to part 1, part 2, part 3, part 4, part 5. Yeah, of course. And then I didn't do a good job of recording the first half, but what I did record, I will post tonight. So I will record everything, or everything that I did record